Hello, everyone. Good afternoon. Thank you for joining today's webinar on scalable 3D human cardiac models for drug discovery. I am Salome and I'll be your host for today. So before we begin, and just to give a few more minutes for people to log in, I would like to share a bit of a few housekeeping items. So this webinar is recorded and will be available on demand within 48 hours of the broadcast. So if you would like the replay, please feel free to reach out. One of our biz developers will follow back with you as soon as possible. And of course, during the webinar, please feel free to ask any questions, send them in the chat, and in the end, we will have a short Q&A session where Silke will try to reply to most of the questions that we'll still have time to. And now with that, I think I have the pleasure to introducing our speaker for today. So with us is Silke Schwenberg, Associate Director, Cardiac Services at Ncyte. She leads Ncyte's cardiac service offerings, and she has more than 20 years of experience in stem cell-based drug discovery. But not only that, but she also serves on the board of the Safety Pharmacology Society. And with this short introduction, I think it's now time to start the webinar. So Silke, I give it back to you.
Hello, everyone, and thank you, Salome, for the introduction. So today I would like to show you our cardiac 3D microtissue model. But before I do so, I would just like to quickly introduce Ncyte to you. So we are a stem cell company based in Leiden in the Netherlands, which is actually a super beautiful city. So if you ever have the chance to come, please come here and visit us. We were founded in 2009, so relatively shortly after the invention of the iPSC technology. And we have a total staff of over 30 people, with a lot of them having like a PhD. And we cover approximately 10 different nationalities right now. We are serving a lot of people and companies within the big pharma, but also smaller biotechs and even academia groups. And we are representing clients across 4 different continents already, and usually have a lot of different projects each year. Our business model is all based on iPSC, and what we have is what we call discovery services that you can think of as being like a contract research organization where we offer certain assays and assay development to our clients. And we also have a cell supply, which is the catalog products that you can just offer off the shelf. So have a look at our website if you're interested in that. We offer safety services, so that means you can send us compound where you would like to see if they have any side effects, either on cardiomyocytes or neurons or any other cell type that we have in-house. And for that, we usually use the standard state-of-the-art assays that everyone else would offer you as well. And then we have our so-called custom iPSC services, where we can use either your in-house IPS lines, or we can generate an IPS line out of patient material and then differentiate these cells into the tissue of or cell type of interest to you. That could be either cardiomyocytes or neurons or endothelial cells or lung cells. So our manufacturing team is super capable of coming up with very good solutions for various cell types. So when it comes to our services, we are really showing the full potential of the iPSC. So as said, in the center, there's always the IPS line. And on the right, that's what I call the custom iPSC services. So that is what is the custom differentiation, generation of custom patient-specific IPS line, or CRISPR editing a disease mutation into these lines. On the left, this is our services. So that means that we use the IPS line that we either have on stock already or that we have made and designed for you. And then there comes the assay development to show phenotypic differences, for instance, between a disease line and a wild-type line, or like a CRISPR-edited repair mutation of a disease line. And then that can go into high-throughput drug screening and drug testing. And that is both for efficacy and then, as I mentioned, also for safety and toxicology applications. So when it comes to 3D cardiac models, which is the topic of today's webinar, there are challenges and opportunities coming with these models. So the 3D models usually should have or allow for a better maturation of the cell types involved, because the environment is more representative of what they should look like also in the patient or in vivo situation. And then they, of course, represent a more holistic tissue model, which applying and integrating the different cell types. And in the case of the cardiac 3D models, we are integrating ventricular cardiomyocytes, vascular endothelial cells, and cardiac fibroblasts. And then as a consequence out of having like all these different cell types in the mix, they should also show a more integrated tissue response. That means that any compound that you throw into the mix will, of course, act on 3 cell types. And then due to the crosstalk of the cell types, the result that you get out of a 3D model might look slightly different from that answer that you would get if you would only investigate one of the cell types in a 2D model. And that brings me already to the flip side. So the challenges with the 3D models, besides usually they might show a higher variability compared to a standard 2D model, and that many of those are limited in their scalability and throughput option. But they ultimately also come with a higher complexity. So that means that if you want to set up such a system, optimizing a system that is a 3D model comprising of 3 different cell types is a bit more complex than trying to standardize and optimize a 2D assay that only has 1 cell type. And then the same is true for the data analysis. So if you generate data from a 3D model, as I was mentioning already, all the 3 cell types will respond to, say, a given compound that you throw on them, but then they might also interact with each other. That means that if you challenge the endothelial cells with a certain drug, they might secrete certain factors that in turn will then activate or deactivate some of the pathways or actions of the cardiomyocytes. So the data analysis and especially the data interpretation is also a bit more complex. The cell types that we use for our so-called heart-in-a-box 3D cardiac microtissue is our ventricular cardiomyocyte, which is the majority of the cells. So 70% of the cells in our microtissues are ventricular cardiomyocyte. As you can see here in this slide, the cells that we produce here at Ncardia have a very high purity, and we usually use a troponin T as the marker for a cardiac identity. And typically, the cells are more than 90% pure of cardiac troponin. In panel B, you can see an immunostaining for cardiac troponin and alpha-actinin. And there you can see that our cells have preserved sarcomere organization. In panel C, you can see an action potential profile that was comes from a patch clamp experiment that was done initially, where you can see that the action potential does mirror that of human ventricular cardiomyocyte. And in graphic number D, that is the compound nifedipine shown in a microelectrode array. And that is actually one of the compounds that we also use for our functional batch release testing. So each and every batch of the cells that we produce here in-house is also functional QC'd before we release the batch. So in this slide, I would just quickly show you the batch consistency and reproducibility. So our ventricular cardiomyocytes are all produced in stir tank bioreactors, and we can produce up to 10 billion assay-ready cells per batch, which means that if you, for instance, would ask us to produce a full batch for you, that would eliminate revalidation across different batches for your own in-house assays. If you look at panel B, that shows the viability and the troponin expression in the cardiomyocyte of 5 different batches across a 5-year span of cryopreservation. So that means the cells were, of course, tested when the batch was originally released and put into the freezer. And then the cells were put out again after 5 years. and then again the viability and the troponin staining was performed. And as you can easily see, both for the troponin content and also the viability, even after 5 years in liquid nitrogen storage, the cells are still healthy and viable, and the troponin expression is still very high. What might be of even more interest is what the data that I show you here in panel C, which is the variability and beat rate consistency across 9 different batches of cardiomyocytes. And what you can see here is that both the inter and intraval variability in our MEA release assay is really low. And also the beat rate, with the exception perhaps of one batch, is clustering very closely together and is very consistent. Our endothelial cells show a very high expression level of both CD144 and CD31, as you can see in the flow cytometry panel number A. And in the image in panel B, that is an image of a so-called acetylated LDL uptake assay, where we prove that our endothelial cells are capable of internalizing the low-density lipoprotein, which is a key characteristic of functional endothelial cells. And I'm not sure how good you can see that, but nearly every single cell that comes with a blue nuclear staining has also the red staining for the acetylated LDL. So what we also did was to test these cells in a classic endothelial barrier assay. So this is a TER measurement. So endothelial, in this case, not epithelial, electrical resistance assay. And what you can see is that with increasing concentrations of this compound, and that was a Klein compound, so that's why it's dubbed compound 12, and I don't have any compound name coming with it. With increasing concentrations of this compound, this barrier was breaking down over time, which means that the resistance, which is displayed on the y-axis, is going down. And then with the very high concentrations, you see that this is not even coming back. In panel D, this was an experiment that was done at our colleagues at Mimetas. You can see a classic sprouting assay in one of their microtissue setups. And here they compared our Ncyte endothelial cells against the gold standard TUVEC primary cells. And what is displayed here is the effect of two tool compounds at various concentrations on these sprouting, so that means like capillary formation of these two different endothelial cells. And as you can easily see, both the TUVEC but also our Ncyte endothelial cells are responding to the compounds, which in both are inhibiting the sprouting of the capillaries. Then last but not least are our cardiac fibroblasts. So the bright field image show that they have a very nice and typical spindle shaped morphology. And then in panel B, you can see that the cells do express both connexin 43 and also the marker vimentin. And in panel C, you can see that despite, in this case, the connexin expression, you can also see that the cells are capable of producing collagen 1 type collagen, which is one of the major components of the extracellular matrix that we need to glue our 3 cell types together in the microtissues. Together with our collaborators at the Leiden University Medical Center here in Leiden, which is literally more all across the parking lot, we now have developed a microtissue model comprising of the 3 cell types. And as I was mentioning already, so we have like 70% of the cardiomyocytes plus 15% each of the endothelial cells and the cardiac fibroblast. And we developed an optimized protocol to form and maintain and label these cells using a robotic system. So we have like a Tecan robot here in-house that we use for this. And all of this is done in 384 well plates. So that means that the formation of the microtissues, the maintenance, the media changes, and then also the loading of the calcium dye is done in the robotic system with minimal like hands-on laborious steps in between. At the day of the assay, which is typically at day 14 after formation of the microtissue, we then load the cells with the calcium dye, and then you do the baseline measurement in your calcium imager, which in our case is an FDSS microcell instrument from Hamamatsu. And then within the instrument, the compounds are injected into the plate, and then you do follow-up measurements, say after 5 minutes, 15 minutes, and 30 minutes to assess the action of these compounds. So in the whole system, when we do that here in-house, we do that in a 384-well plate format, which allows us to run a decent number of compound with full dose response curves. And if you think about a more basic screen, just perhaps testing on 1 or 2 concentrations, you can literally really run like up to 1,000 compounds per day. The 3 cell types are pre-cultured individually because they all need their own specific medium. And then at the day of the formation of the microtissues, we dissociate them, and then it goes into the Tecan robot for the formation of the microtissues. So when the model was developed here at Ncardia originally, my colleagues at that time did some cell titration testing. So here on this slide, what you can see is the some representative images of the total cell number representing 20,000, 10,000, and 5,000 cells. And in this case, we tested 3 different batches of the cardiomyocytes. And in this case, it was wild-type cardiomyocytes. But we used the same batch for endothelial cells and cardiac fibroblast. And what you can see here in these representative images is that with all 3 batches of cardiomyocyte, we were able to form the microtissues. And actually across the different days, so here on the x-axis, so you have like the different batches of the cardiomyocytes, like batch 1, batch 2, batch 3, and then each of the graph colors or bar colors represents one a day after the tissue formation. So we have day 1, day 7, and day 14. And then with the different seeding densities, total cell number from 20 to 10 to 5,000 cells, you can see that yes, there is a big gap from the 20,000 going down to the 10, but then in terms of the size of the microtissue, the difference between the 10,000 and the 5,000 is less pronounced. And what is even more important is that the variability across all the microtissues here from one day and one seeding density is really low. And also they do not really change a lot over time. So that means that we do not see, say, an explosion of the cardiac fibroblast population, and then the microtissues become bigger and bigger. But we also do not necessarily see any necrosis, shrinking, or whatever of the microtissues. So they are quite stable during the assay time that we tested them so far, and they are alive and happily beating in their plates. So, and then the image on the right, upper right part here, that is actually a full 384-well plate at day 7 post microtissue formation, where you can see that using the Tecan robot in each of the wells, you have 1 microtissue. Usually you really only get 1. You might see some debris or singular cells sitting around it, but we do not typically get like multi-microtissues or whatsoever. So this is really a very robust and easy to use methodology. So when we set up the assays, we also challenged the microtissues with a couple of tool compounds. So in this case, we used isoprenaline, nifedipine, and then BK8644. And what you can see here is that, as expected, with isoprenaline, you do see an increase in the beat rate. And this here is just like the calculation out of that. So you see that's highly statistically significant. And if you look here at the original trace, you can easily see that after injecting the compound, indeed, the beat rate is, or you have more peaks in the same time frame. But what was very interesting is that we also could see an increase in the amplitude. So that means that the contraction force should be bigger with an increase in the isoprenaline concentration. What you can also very easily see is if you add the calcium channel blocker nifedipine, there is not really any difference in the beat rate, but of course the calcium transient amplitude is depressed, and this is exactly what you would expect. So here in this case, you can see that towards the end of the incubation period here, that the amplitude height is getting smaller. And the opposite happens with the third compound, BK8644, which is a calcium channel activator, where you can see, again, not a lot of change in the beat rate, but there is an increase in the amplitude of the calcium transient that we measured. What we also tested is if you could use the cardiac microtissues to study cardiac fibrosis. And this was really, say, like a shotgun experiment, just one try and check what we can get out of it. So in this particular case, we challenged the microtissues with increasing concentrations of TGF-beta, which is a key mediator of cardiac fibrosis, which would then promote extracellular matrix deposition and also cardiomyocyte hypertrophy. And one of the best markers to measure cardiomyocyte hypertrophy is actually the release of NT-proBNP. And that's exactly what we did here in this experiment. So the bar graph on the right shows you on the y-axis the different concentrations of TGF-beta, and as a positive control, we used endothelin-1 because endothelin-1 is like the model inductor of cardiac hypertrophy. And indeed, with endothelin-1, you get really huge amounts of NT-proBNP released into the supernatant. But what you can also see is that with these increasing concentrations of TGF-beta, so from the left to the right is the concentration increase, you do also see an increase in the release of NT-proBNP. So that means that obviously TGF-beta is triggering kind of a hypertrophy response in the cardiac microtissues, and that would be also one of the steps in developing cardiac fibrosis. So now our collaborators from the LUMC, which is in this case the group of Richard Davis, they are particularly interested in using 3D cardiac microtissues as disease models. So when you generate an IPS line from a patient with a particular genetic disease, in the best case scenario, the differentiated cells, and it doesn't really matter if this is like the cardiomyocyte or the endothelial cells or the cardiac fibroblast, they all should reflect the phenotype of the disease pathophysiology, and in the best case scenario, even that of this exact patient. So you know that with each patient for a certain disease, the symptoms that they have, the severity of the symptoms, how exactly they present the symptoms might differ. And in the best case scenario, if you generate an iPSC model of two patients with a certain genetic or inherited cardiac disease, there might be slight differences in how these patients present the symptoms. And very often you would also see differences in the given iPSC model that the phenotype might not look exactly the same between the models derived from the two different patients. Another option to induce or to generate a disease model is, of course, to use CRISPR editing. And this is also something that you can easily do. So in that case, you would use apparently healthy or wild-type iPS line, and then you would CRISPR edit a disease mutation into this line. And then hopefully you will see that that would translate also in differences in the phenotype of the cells. The latter option has the beauty that it comes with its own isogenic control because the wild-type line would serve as such. The limitation of the CRISPR editing is that this would not be a good basis if you think about a personalized medicine approach, because for that you would really have to start from the patient material. So in the current collaboration that we have with the LUMC, the disease of interest was CPVT1. And CPVT stands for catecholaminergic polymorphic ventricular tachycardia. So this is really a tongue-breaking disease name, which is an inherited channelopathy with arrhythmias triggered during adrenergic stress, for instance, during exercise phases. And in this case, the adrenergic stress would increase the calcium in the sarcoplasmatic reticulum, which would then end in spontaneous calcium leakage and arrhythmias. CPVT patients harbor a ryanodine receptor mutation, which then in turn also leads to a leaking ryanodine receptors. And that is then also increasing the calcium levels and also lead under stress situation to arrhythmias. So the goal of the project that we had with the LUMC was to generate a robust arrhythmia model for CPVT1 patient in cardiac microtissues. Then to automate a 3D model formation and the analysis pipeline. And then as a last step, perform a high throughput screening to identify novel antiarrhythmic compounds, or at least compounds that have not been formally known in the context of the CPVT disease. So we were successful in generating the cell types from CPVT mutant line. And as I showed you, we were also very successful in generating like an automated 3D microtissue formation. The LUMC in turn also came up with an automated way to analyze the data. So what you can see here on this slide on the right hand is calcium transients of the CPVT microtissues. So the upper panel shows you the background, very regular beating of the microtissues. And then when you add a certain trigger that is mimicking the stress situation in the patient, you can see a very pronounced arrhythmic signal of the microtissues. And then when you add the drug flecainide, which is used also in patients to rescue the arrhythmias, you can see that the calcium transient signal is going back to a normal signal. And then when you translate that into numbers, and this is what we call like an arrhythmia score, in the percentage of the microtissues that show arrhythmia without adding the trigger is really small. But then when you add the trigger, approximately 90% of the cardiac microtissues show a phenotype. And then when you add the flecainide, they are rescued from that. If you only add the vehicle of the flecainide, which in this case is DMSO, there is also a certain variation between the ones with only the trigger and ones with the trigger plus DMSO, but it's nowhere close to what you can rescue then with the flecainide. So for us, that was the proof that the model as such is showing the pathophysiology that you would also see in a patient. And that then triggered the next step, which was a pilot screen of 95 FDA-approved compounds. So that were not compounds specifically approved for the disease itself, but it were all marketed drugs spanning various mode of actions, so drugs acting on calcium channels, on sodium channels, and they were all in the market already. So the LUMC came up with a rescue score, and everything above an average of 44% rescue was considered as a hit in this screen. And out of these 95 compounds that were initially tested at 3 different concentrations, 17 compounds were identified as a hit. And 9 of these compounds have not been tested in CPVT1 patients so far. So that was already very promising, because we could identify compounds where there was no prior knowledge that they might be good in rescuing the CPVT1 phenotype. The real compound screen was then including 2,000 compounds from the SPECS repurposing library. The screen was performed in the fully automated workflow that I introduced in one of the previous slides. Throughout the screen, we did not observe any microtissue loss due to the handling. And we were able to screen the 2,000 compounds in triplicates within 2 days. The per plate QC confirmed the expected response to the positive controls. And across the screen, we had a variability of less than 10% in the microtissue size. And the average Z prime factor was 0.6 across the entire screen, which is really good. Some of the key outcomes was that we were able to produce sufficiently large numbers of all 3 cell types, as well as sufficiently numbers of microtissues to validate and support all phases of the screening cascade. We could identify compounds that were not previously evaluated in the CPVT patient model or in clinical studies. And what we also achieved was that there were some very interesting hits out of the 2000. So the total hit rate was 4.6%. So 97 compounds identified out of the 2000 that were tested. So the modalities, or let's say the disease areas that were covered within the hits was rather broad. And in the next slide, I show you that we had some disease or application areas where we found an increased number in hits, and the hit rate was especially enriched in neurological, cardiac, anti-inflammatory, and antiviral categories. So the graph on the right, on the left y-axis, that is the total compound number falling into this specific class, so out of the 2000 compounds. And then on the right, that is the compound hits that falls within this class. And then on top of that is the percentage or hit rate percentage of that particular class. And as you can easily see, the biggest hit rate was coming from the neurological field, followed then by the cardiovascular, which perhaps is not that much of a surprise, anti-inflammatory, and then also antiviral agents. And in the meantime, the LUMC already retested a subset of the compounds, run additional compound tests to verify the hits, and I'm already very curious to see the results of the latest experiments that were performed. So the key takeaways are that the Ncyte heart-in-a-box cardiac microtissue model can be used at scale in a high-throughput screen. The microtissues require really low number of total cells and are very homogeneous with low variability in terms of size. At Ncardia, the model can be used to generate approximately 1,500 data points per day based on calcium transient as a functional readout. The setup can also be applied to other iPSC-based cardiac disease models to study phenotype rescue. And of course, the model is also suitable for the assessment of cardiac safety and also especially of structural cardiotoxicity, because we do have the 3 cell types in the mix, especially when it comes to structural cardiotoxicity. That might be a very interesting model to be used. And with that, I will give back and hand over to Salome for the Q&A.
Thank you, Silke, for the lovely presentation. There are a couple of questions that came in already. If you have more, please share them in the chat. So if Silke, you can come back on stage and I can direct them to you. So the first question: you have demonstrated very nicely a physiologically relevant disease assay for CPVT. Is this assay also suitable for other cardiac diseases?
Yeah, absolutely. So everything where you think that either all 3 cell types might be necessary to show a good phenotype, or where you think that a 3D model would be superior over a classic 2D 1 cell type only model, this would be a suitable approach. So we do not have any disease model cell types on stock that we could routinely use for that. But as I was mentioning at the very beginning, Ncardia offers a custom manufacturing. So if you have a specific disease mutation in mind, or you might even have already an iPSC line, either derived from a patient or coming with a CRISPR mutation, we could differentiate the cells from that and make the microtissues. And then we would have to discuss what would be the suitable readout, what are some tool compounds that could be used to demonstrate efficacy. But that is definitely something that is worthwhile to look into.
Okay, thank you. I'm going to follow up with the next one. I'm taking them in the order that they came in. So how is Ncardia's heart-in-a-box microtissue model compared to other 3D cardiac models like EHTs?
That's a good one. So I think that our model has the beauty of being relatively simple, and we get to a good throughput. So what I perhaps forgot to mention is when we assemble our microtissues, we do not have to add any extracellular matrix. So we just mix the 3 cell types, we put them into ultra-low attachment, U-shaped plates, and then they assemble themselves into the microtissue. So what seems to happen is that once the 3 cell types come in contact, and then say the cardiac fibroblasts also start to secrete some extracellular matrix proteins, the 3 cell types really glue together perfectly to form these microtissues. So it's a relatively, let's say, simple setup, because you really don't have to add any other matrix proteins or whatever to the mix. And the other advantage is that you can really run it very easily, or even preferentially, in the 384 well plates, which really allows for a decent throughput. A classic EHT, of course, comes with a lot of other advantages. So there you usually can apply a certain stretch to the cells. You have like these little posts that hold the EHT that you can build in certain stiffnesses, which also might further force the maturation into where you would like to go with these models. But it's also way more complex. You usually need more time, way higher cell numbers for many of these EHT models, because they usually come in, say, lower well sizes. So say something like 12 well plates, 24 well plates, perhaps 48 well plate format. That means that you also need substantially more cell numbers for the EHTs. From screening cascade thinking, I would think that a relatively simple microtissue model like our model would be better for a real screening, and an EHT model might be better for more in-depth mechanistic studies, because there you can really also play around with other factors that we can't model in our like spheroid organoid system.
Okay. If I would also like to add here that if you want more additional input on the questions, please feel free to email Silke. I'll email you her email after the end of the session. And with that, I think we're going to go with the last question, which is a pretty long one. So Silke, you might want to take a look at it yourself as well. So with the integration of iPSC-based models in high-throughput screenings, what steps are taken to minimize variability across different platforms or conditions during long-term studies?
So I assume that means you use the same cells in different assays, like say you want to compare from an MEA recording to our microtissue model. So at Ncardia, what we are usually doing is to have certain assays that we use as a batch release assay. As I was mentioning initially, for our cardiomyocytes, for instance, that is a classic MEA assay. But of course, what we are not necessarily doing is to test the cells in all possible assay systems that you can think of. So that means whenever you really want to compare across different platforms using the same cell model, that would be something where I think you should use certain model compounds and then really test them, say, in an MEA versus the 3D heart-in-a-box model. And then you should look at the data and then try to interpret that in a way if the assay would show the results that are supposed to be seen. And then you really have to look into the variability across the different platforms. Coming from the cell manufacturer perspective, what we are usually aiming for is to really have a very low variability across the batches that we produce. And I think I could demonstrate that with one of the earlier slides, both with the troponin expression levels, but then also with the functional MEA data across several batches. And then this would also hold true for the other cell types that we produce here at Ncardia. But I understand where the question comes from. So this is really something that is important. Where it gets a bit more complicated is, of course, if you look at models provided by different manufacturers. So there, it might be a bit harder to harmonize that from one cardiomyocyte vendor, cardiomyocytes to the next one. But what I learned is that very often, even if the base parameters might not be all identical, say cell type A has a higher or lower beat rate, for instance, or needs more or less culture time to get to a certain state. Very often, if you look at the pharmacology, that is still really very representative across all the different cell providers batches.
Nice. Thank you, Silke. This was the last question for today. Again, thank you all for taking the time to join today's webinar. If you have any more questions, and to those questions that we didn't have the time to answer live, Silke will follow up with you. I want to remind again that the webinar is recorded and the replay will be sent to you. Please let us know if you would like to receive it. And with that, see you till next time. Good afternoon.